Hello everyone, we are from Group 6 and we'll be presenting about genetic improvement in salmon. So this is an example of the types of salmon that can be found around the world. Our group consists of me, Shifaya, Sri Vayoni, Pratasha, Weiching, and Sun. So, salmon is a common name for several species of the family Salmonidae. Salmon live along the coast of both North Atlantic and Pacific Oceans. Salmons are anadromous, which means they lay their eggs in fresh there for several years before moving to the ocean. So this is the taxonomy of salmons and some information about their chromosome number. Background. The farming of Atlantic salmon has become an important industry in several countries, and breeding programs have been implemented to improve genetic performance and adaptation to farming environments. So, seven different traits, which are body weight at slaughter, age of sexual maturation, survival in challenge tests with furunculosis, flesh color, total fat content, and amount of fat tissues have been included in the breeding goals. So these are the main traits that the breeding goals normally focus on. So salmon families usually have high resistance to IPN, but IPN, which is infectious pancreatic necrosis, is a highly contagious systemic burnovirus disease of young fish of salmonid species held under intensive rearing condition. So under intensive rearing condition, this disease can be highly contagious, but normally salmon families have a high resistance toward it. And the aim of our program is to introduce high resistance towards IPN in salmon under intensive rearing conditions. So experimental design will be explained by Sun and Sri.
So for this part, this one is for the breeding. So first thing we need to know that the two genome-wide significant QTL for IPN is in the chromosome 21 and also chromosome 4. So at least two full sibling families of salmon fish are bred. Okay, these are the two types of mating design that can be used, which are the hierarchical mating design and also partial factorial mating design. So for the first one, each male fertilized 2 to 3 females, and for the partial factorial mating design, eggs from each female split into 2 and fertilized by 2 different males. So the offspring with homozygous dominant Q for IPN resistant gene is in chromosome 21 or chromosome 4 will be selected and also further bred. Next.
So the first step is to identify the linkage, and the second one to find the co-segregating markers. As genetic markers near large numbers of sites throughout the genome, and examine to find a site close enough to the disease-causing site to appear associated with the disease, and we usually use SNPs as the markers.
So the next one is we need to develop yacht contig, or also known as yeast artificial chromosome. So basically this is used to clone DNA sequence in yeast cell, and the yeast will grow and divide so they can amplify the yacht DNA, which we can use that for the DNA mapping and also for the sequencing. So we use yacht contig instead of BAC, which BAC, using the bacteria, because the genome size is big. Many genes is controlling the IPN resistance. The next step is we use ORF to recognize the gene of interest, because ORF is the sequence that probably have high possibility to contain the gene that we are looking for. So in best situation, only single ORF is found. However, this is not the normal case. This is not the often occurred. So if we found several RF, we need to introduce candidate gene into the genotype that is mutant for the gene by genetic transformation. This genetic transformation can be done simply like in the plant. However, for this experiment using animal, we can also use the mutational analysis, which we need to compare the ORF with the DNA from the library. So basically, ORF are used as probe to select complementary from library containing mutant DNA. And the ORF also compared with the library of the normal DNA. So ORF from mutant library, which is different from its counterpart in the normal library, has the strong, it will be the strong candidate for the gene. So meaning we can spot the gene on the candidate. Next.
As NCBI, we can find some genes which have been studied by the scientists. So we need to find the LD between the SNPs in the database and the new sequences. As we can see in the picture, if the new SNPs have high LD degree with the SNPs, so we can determine the area of the new SNPs which contributed to the IPN disease. Next one. We use software to calculate the number of SNPs based on the effective number of chromosome sequences. We suggest that at least 74,000 SNPs will be necessary for an association mapping study and genomic prediction. Next one. We can see that LD declines mostly as the physical distance increases between markers. As we can see in the figure, maximum average LD of 0.21 was estimated for SNPs less than 1 gigaparsec. This value declines quickly as the marker distance up to 0.1 MB with a value of 0.16. From 1 MB to 10 MB, LD range from 0.14 to 0.07. The latter value represents the lowest average LD estimated in the current data set. Next.
Hi everyone, I'm Pratasha and I'll be talking about the QTL mapping. So the aim of QTL mapping is to detect which regions in the quantitative trait locus of the genome affects the certain trait that we're looking for, and to also describe the effect of QTL on the trait. There are several Atlantic salmon genetic linkage maps currently existing. Composite map for salmon combines the molecular marker and mapping data from the genomic research. There is data available from 3 areas of research, the Atlantic Salmon Project, Salmon Genome Project, and EU Collaborative SALMAP Project. Next.
For selective breeding for QTL-X, first genetic markers was used to select the desirable eggs, and the desirable eggs here is egg with high resistance to the viral disease IPN, the infectious pancreatic necrosis, and this stress is correlated with high degree of robustness and thereby reduction of loss throughout production cycle. And we can also determine whether individuals have 0, 1, or 2 variant copies of allele by directly selecting the IPN resistant fish. And the theory involved here is the infective group plus genetic testing equal to greater flexibility in product character. Next. So for the diagram here is shown protection against IPN of different number of dominant Q in the parents. We can see that if there is 0 dominant Q in parents, then the protection against IPS is 0% and is also a control group. And if the number of dominant Q is 3, then the protection against IPN is as high as 80%. Next. For the conclusion part, Linkage disequilibrium LD approach is beneficial to detect the linker genes that govern the threat for IPN. So by determining the genes linkage and location through SNP, this will help researchers to detect which genes governing the threat IPN. So that's all from our group. Thank you.